so that's where we're going to pick up here today. So we talked about how phospholipids are these sort of schizophrenic molecules. You know, they have a hydrophilic end or a water-loving end, and they have a hydrophobic end or a non-polar end. So in the cell, they arrange themselves in this bilayer. So the heads are all pointing out, and the tails are all pointing towards each other. And if you take that and make a sphere out of it, oh, here, I got to start my thing. There. If you make a sphere out of it, you get something like this, and this is essentially the cell membrane, the nuclear membrane. This is also the structure of the membranous organelles that we're going to talk about later, like the endoplasmic reticulum, the Golgi apparatus. The mitochondria also has a bilayer membrane like this. So for the most part, wherever you hear the word membrane in the cell, this is really what we're talking about, is a lipid bilayer that has formed a kind of fluid, flat structure. So in addition to the phospholipids, which make up the bulk of the membrane, we also have a number of other important structures that are found in the cell membrane as well. You can see these kind of strange yellow molecules here. Those are cholesterol. You know, we hear a lot about the downside or the bad things about cholesterol, but we do actually need cholesterol. And a lot of the cholesterol in our bodies actually lives in between the phospholipid heads. So in this non-polar region in the center of the membrane. And cholesterol helps to keep the cell membrane very flexible and fluid. So if you were to totally eliminate cholesterol from your diet, you would actually get into trouble eventually, although the body can make it, because cholesterol is a necessary component of the cell membrane. So those are these yellow blobs in here. But then the more obvious things are these giant structures here and here and here that are either sticking through the membrane or sitting on top of it. And those are membrane proteins. So our current best understanding of the cell membrane is that it's a little bit like an ocean that has stuff floating around in it. So you have to think that the cell membrane is very fluid and flexible. You know, these phospholipid heads are not bound to each other. You know, so this protein right here, for example, can float around inside this membrane, kind of like an iceberg in the ocean. So we call that the fluid mosaic model of the cell membrane. And it basically describes that things can move around in the membrane very easily, which is important because, as we'll see over the course, these membrane proteins change and do stuff all the time. So it's important that they be able to move around, and you know, the cell can pull one out and put a new one in, for example. And all of that is possible because of the fluidity of this. So these are what we call integral proteins, or proteins that either are embedded in the cell membrane or pass all the way through the cell membrane. So for some examples of those, the big classes of membrane proteins, we have anchoring proteins, and these attach the plasma membrane to something else. So, for example, other cells. You know, the cells of our skin have to be tightly bound to each other so that they don't come apart and so that they can provide the barrier that the skin needs to provide. So it's anchoring proteins that do that. You know, you've got a protein on the surface of one cell like this, and then you've got a protein on the surface of its neighbor cell, and the proteins might hook around each other or interact or connect somehow. So that would be anchoring proteins. Recognition proteins, the cell membrane is covered in these proteins that are essentially like name tags. We're going to learn, you'll learn about the immune system next semester, but one of the things the immune system has to do is destroy anything that doesn't belong. Well, in order to do that, the immune system has to be able to tell what does belong. So these recognition proteins are present on all cells, and they're sort of like a name tag for that cell to the immune system. You know, it says, I'm a normal, epithelial cell, or I'm a normal muscle cell. So these recognition proteins help the immune system to identify what kind of cell is present. We don't talk very much about these first two, but these last three we talk a lot about. So receptor proteins are proteins that bind to a chemical or a molecule that's found in the extracellular fluid. And when they do, they create some change on the inside of the cell. So hormones, for example, like insulin. Say this is a receptor protein right here, and insulin binds to this spot here. That binding creates a change on this end of the protein, which sends a signal inside the cell to change something or do something different. So a receptor protein is kind of like a switch that is thrown on the outside of the cell that actually makes something change on the inside. And then these last two are ways for substances to get from the extracellular fluid into the intracellular fluid or vice versa. So we have carrier proteins, which we're going to talk more about later on in this chapter, that basically shuffle molecules from one side of the membrane to another. And then we have channel proteins, like this one here in the center, that have a consistent opening all the time, but only for either one or a very small number of molecules. So, for example, there are water channels that allow only water through, nothing else. So these channels are often very specific, but they're open all the time. So the difference between a carrier and a channel is a carrier opens and closes, and a channel is open all the time. So we'll see more examples of these as we get into the organ systems. Like we're going to talk a lot about channels when we talk about the nervous system, and we're going to talk about receptors when we talk about the muscle system, and so on. All right, so just an introduction to membrane proteins. So in addition to proteins on the membrane, there are also carbohydrates. So you can see these tree-like things that are sticking out. So here's one, and here's one, and here's one. Well, each one of those is a sugar, and those sugars have been bound together. We talked about polysaccharides. I think it was the time before last. Well, this is an example of a sugar that's kind of been all strung together, but it's attached to the outside of the cell membrane. So when we zoom in real close to a membrane, you can see this fuzzy stuff right here. These are those carbohydrates that are sticking out. So we call that the glycocalyx, and it provides a kind of soft, slippery shell around the whole cell outside of the membrane. Now, in plants and many bacteria, the glycocalyx is very advanced, so advanced that we call that the cell wall. But, animal cells like ours don't have a cell wall, but they still have this glycocalyx that performs a very similar function. So the carbohydrates on the surface, they help to lubricate and protect. They help to hold things together or to allow things to move. And then they're also important in recognition of, one cell recognizing another. So the way I think of the glycocalyx is, if you can imagine the cell membrane, they're like trees that stick up from the top of the cell membrane and create a kind of canopy over the top of it. So those are membrane carbohydrates. Okay, did everybody answer the last name question, I hope? Because I'm going to close it out. Looks like two of you haven't. If you haven't put your last name in, please do that real quick. All right, because I'm going to end it. 25. There we go. There's everybody. Okay, now we can go to the next question. All right. So which of those things is true about the plasma membrane? And you'll get your question here in a minute. Multiple choice. There we go. Did I hit the wrong thing? I hit the wrong thing. Fat fingered it. There, now you should have the right one. I'll give you extra time. Give it a second. It is not a perfect system. But it's what we have to work with. I'm getting answers, and did it change? Did it change or no?
Yes.
If it didn't, just wait till the next question. All right. All right, I'm going to close it out so we can go on. Okay, so the plasma membrane. So A, separates the nucleus from the rest of the cell. There is a membrane that does that, but it's not the plasma membrane. That's the nuclear membrane, which we'll talk a little bit about more in a minute. Now, the nuclear membrane and the plasma membrane are, virtually identical, but it's what they surround that matters. It's not rigid. Remember, we talked about it's very fluid and flexible. It is permeable and variably permeable. In other words, the cell can change the permeability of the membrane. It can allow more things in or less things in as needed. And it has 2 layers of phospholipids, and absolutely E is one of the primary functions of the plasma membrane, and that is to regulate the movement of materials into and out of the cell. So just like we as a whole organism pay a lot of attention to what we put into us and what comes out of us, the cell membrane is doing that same thing. You have to control the inputs and outputs in order to maintain homeostasis. All right. So from the plasma membrane, we go inside the cell, and we're going to talk, for a lot of this chapter anyway, we're going to review the organelles. Now, I know all of you have had some version of cell biology, either in high school or college. So we're going to go kind of quickly. We're not going to spend a lot of time on each of these organelles. It's more just a refresher than anything else. The cytosol is also called the intracellular fluid. So this is the fluid that the organelles and the other structures inside the cell are surrounded by. So, and like I said last time, the ICF is significantly different than the ECF, or the extracellular fluid, that fluid on the outside of cells. So 2 key differences, sodium and potassium. Inside the cell, potassium levels are very high, sodium levels are very low. Outside the cell, it's the opposite. Potassium levels are very low, sodium levels are very high. That's an important difference that we're going to see play a role in several different key spots in physiology. And then another big difference is in protein concentration. The inside of cells in the intracellular fluid has a ton of protein in it. Those are enzymes, structural proteins, components of organelles, all different kinds of proteins doing all different kinds of things. And while there is quite a bit of protein in extracellular fluid, it's not nearly as concentrated as the intracellular fluid. So that's another key difference between the two, is the amount of protein that's present. Outside the cell, it's mostly sodium and chloride. Inside the cell, it's mostly potassium and proteins. All right. So cells are not, a lot of times we think of cells as kind of these bags of water, these complicated balloon-like things that you could push on and they would just, change shape however you want. But the truth is they have a skeleton, just like, we as a human being have a skeleton. And it's made of different kinds of filaments and parts. Now, the cytoskeleton does not come up very often in the course, so I'm not going to spend a lot of time here, but just know that there are different kinds of filaments, and they are what give the cell its shape. So when you look under the microscope in lab, and you see that a cell has a tall, skinny shape, it's the cytoskeleton that has done that. It's not like it's been shaped from the outside. It shapes itself from the inside with these tiny protein strands that give it a sort of rigid structure. All right. Some cells have kind of an advanced design like this. Microvilli are these little hair-like projections that stick up from the top here. Typically, we see microvilli on cells in the digestive tract and anywhere where we have to see absorption or excretion. And the reason for that is these microvilli expand the cell membrane. If this cell were smooth, there would be much less membrane than all of these little nooks and crannies that we see going up and down the microvilli. So microvilli are present to increase the surface area of a cell. So we're going to see these particularly in the digestive tract, and we'll talk more about why that's important when we get there. All right. The cytoskeleton, one of the key parts of it is called the centriole. In many ways, the centriole is the kind of center of the cell's skeleton. And it's important because in cells that divide, it's the centriole that really guides that division, turning one big cell into two identical smaller cells. So it's the hub that where mitosis is controlled, is the centriole. So cells that do not divide, like muscle cells and nerve cells, they don't have centrioles because there's no need for them. So centrioles and then cilia are a little bit like microvilli, but microvilli don't move. Cilia move. They move in this kind of, so you can see a picture here. It's kind of like rowing a boat. There's a power stroke, and then you lift up the oar and move it forward so you can do that again. Well, the cilia kind of do that same thing. There's a rigid power stroke like this, and then there's a return stroke where it bends back down. We see cilia in a couple of different places. We see them in the respiratory epithelium, which we'll talk more about next semester. But basically, the cilia help to keep mucus moving through the respiratory tract. And we also see cilia, they make up the tail of the sperm cell as well. So both of these structures are made up of microtubules, and microtubules are the largest of the cytoskeleton components. Okay, so centrioles and cilia. All right. Ribosomes are protein-making machines. So we're going to see here in a minute that DNA contains the recipe for creating proteins, and that recipe is turned into a protein here at the ribosome. So the ribosome uses an RNA template to create a polypeptide chain, or a protein, a long string of amino acids. So we see ribosomes in two places. We see them scattered throughout the cytosol. We call those free ribosomes because they're not attached to anything. And then we also see fixed ribosomes as part of the rough endoplasmic reticulum, which I'm going to show you here in a moment. But wherever ribosomes are present, proteins are being created because that's essentially what the ribosome does. It has two subunits, and when it's actively busy, the two subunits are attached together. When it's not busy, they are not as firmly attached. So essentially, the two subunits kind of clasp around that RNA molecule that we'll see later. Okay. Endoplasmic reticulum, very big long name, but a pretty simple organelle really. All the endoplasmic reticulum is a network of membranes. So, you can kind of see here that, here's a membrane, and it's walling off this sort of complex three-dimensional structure around the nucleus. Well, that's all the endoplasmic reticulum really is. It's a convoluted, all twisted up, bent up organization of membrane. Now, the reason that we have endoplasmic reticulum is so that this space in here that is inside the ER can be made different environmentally than the cytosol itself, which is out here. And the reason that that's important is in order for proteins to end up in their right shape, and remember we said it's all about shape, the protein has to get to be the right shape or it doesn't work. In order to get to that shape, the cell has to control what's around that protein as it's being made. So essentially, the endoplasmic reticulum allows the cell to have lots of different rooms that it can change the environment in. So it can change the pH, it can change the salt level and the potassium level and the calcium level. It can alter all those things so that that room or that area of the endoplasmic reticulum is just perfect for the protein that's being made. All right. So we have two different kinds of ER. Rough endoplasmic reticulum, which is what you see here. You see all these little dots on it, and under the microscope, you can see these little dots. Those are ribosomes. So the rough ER is the site of protein manufacturing. Both kinds of endoplasmic reticulum are sort of like factory sites. They're places where things are created. The rough ER, what's being created are proteins. Those proteins are eventually packaged and then transported to the Golgi apparatus, which I'll show you in a minute, to finish developing. The smooth ER is just like the rough ER, only there's no ribosomes. So no ribosomes means no protein synthesis. So the smooth ER is still the site of production of lots of things, but not proteins. So you can kind of think everything but proteins happens in the smooth ER. So we have our phospholipids and cholesterol that are part of the cell membrane. We have steroid hormones, which are important chemical messengers. We have triglycerides, which we use as our energy storage molecule. You know, it becomes adipose tissue storing energy for later. And then also glycogen, which we talked about a couple days ago. Glycogen being branching chains of glucose molecules, another energy storage molecule. So all of those things are formed inside the smooth endoplasmic reticulum. So if we zoom in to one spot of the rough ER, we can see how a protein ends up being made in here. So here you have your ribosome. It's kind of on the outside. You know, you might think it should be on the inside, but it's not. It's on the outside. But then as the protein is made, the protein kind of is made into the endoplasmic reticulum space, which is in here. So we've got our RNA, we've got our growing protein. You know, eventually translation continues, which we'll talk more about in a minute, and we get this, you know, globular protein that a single strand is kind of starting to twist up. Eventually, this protein is done, so now these are complete. Things might be added to them in the endoplasmic reticulum. That does happen. Like here, sugars have been added to this protein to make what's called a glycoprotein. And then eventually the protein is packaged in a little bubble of membrane that sort of bubbles off the endoplasmic reticulum. And then it can separate here to form what's called a transport vesicle. A vesicle is just a bubble of membrane. And a vesicle can contain all different kinds of things. So it's not really an organelle. It's simpler than that. It really is just a bubble of membrane. So you can imagine if this little spot right here were to pinch closed, we'd end up with a bubble that had protein inside it. So that would be a vesicle, and then this vesicle would head to the Golgi apparatus so that these proteins could be finished. Because what's in here, you know, the proteins have been all put together, but they're not in the right shape yet. So some of that has taken place in the endoplasmic reticulum, but proteins take on their final shape in the Golgi apparatus, which we'll talk about next. So the endoplasmic reticulum is all membranes, right? But if you look carefully at it, all of the membrane spaces are connected together. You know, so you can always get from one room to another without going through the cytosol. Does that kind of make sense? Where the Golgi apparatus is also membranous, in other words, it's a collection of membranes, but here they're individual membrane sacs that are kind of stacked on top of each other. So the classic description of a Golgi apparatus is it's like a stack of pancakes. You know, you have a membrane sac here, then you have another separate one underneath, and so on, and so on, and so on. And the role of the Golgi apparatus is to take the proteins that were made in the rough ER and get them into their final shape. Because once they're in their final shape, now they're functional. They can actually do what they're supposed to do. So the way that the Golgi works is it receives proteins from the endoplasmic reticulum, which is down here, through those transport vesicles like we talked about. So those vesicles join up with the first pancake, so to speak, and they dump their contents into here. Now, in each of these membrane sacs, the environment is a little different. You know, so the pH is a little different, the calcium, you know, all of the chemical environment is different in each one. So the proteins go from one sac to the next in those vesicles again until they get all the way up to the top pancake, and by then they're finished. You know, so they have attained their final shape, and now they're functional. So we call that the maturing phase of the Golgi apparatus. In other words, it's the end of the assembly line. You know, if the amino acids were all put together in the endoplasmic reticulum, the final shape takes place as it passes through these different stations in the Golgi, and then the Golgi release those newly formed proteins. They're put back into vesicles, and now they go to the cell membrane and are released. So in nerve cells, for example, neurotransmitters follow this pattern. You know, so eventually they're brought to the cell surface, and then they're released into the extracellular fluid. Now, the Golgi participates in two other things as well. One is called membrane renewal. You can imagine that The cell membrane, has to be constantly replaced. Those phospholipids everything wears out. Just like everything in your house or car or whatever eventually wears out. Same thing happens biologically. Stuff wears out. So it has to be replaced all the time. So the Golgi plays a role in that. It releases empty vesicles out to the cell membrane, not to move anything other than membrane itself. So, cell membrane is made down here and then moved out to the cell membrane to replace older or broken parts. And then another thing that the Golgi participates in is to create lysosomes. Lysosomes are vesicles that have digestive enzymes in them. In other words, enzymes that destroy stuff. So we see those three things. But the primary role of the Golgi apparatus is to finish protein production, to create proteins in their right final shape. All right. So secretory vesicles are just membrane bubbles that contain something that are going to be dumped into the extracellular fluid space. So from the Golgi apparatus, those final completed proteins are packaged. The vesicles are moved out to the cell membrane. They move along the cytoskeleton, so it's not like they're floating around. They actually are transported almost like a car on a road. They're transported along the cytoskeleton, and then when they get to the membrane, those things are released. All right. So like I said, lysosomes are vesicles that contain digestive enzymes. They really have three primary functions. One is to destroy organelles. Just like the plasma membrane, organelles wear out. And, old or damaged endoplasmic reticulum or mitochondria or any other organelle have to be destroyed so that their nutrients can be recycled. So we see lysosomes doing that. It'll surround a mitochondria, and then the mitochondria gets destroyed, and then what's left gets dumped out into the extracellular fluid space to be gotten rid of. We also see lysosomes in phagocytosis, or cell eating. The cell has taken something in from the outside. Let's say it's a bacteria. And the lysosome joins with this, busts it up, and then again dumps it back out into the extracellular fluid space. And then this is the worst case scenario. When lysosomes break open, their digestive enzymes get spilled into the cytoplasm, and they can end up digesting all kinds of stuff there. So one of the things that happens in cell death, when an old cell dies, is the lysosomes inside start to break open, and essentially the cell digests itself. So it destroys itself from the inside. So those are the three primary functions of the lysosome. All right. So ribosomes are organelles that are responsible for which of those things? Have you tried refreshing it? Another trick is to log out and log back in. It's not supposed to do that, but. And another thing is the Chrome browser tends to work better than some of the others. So that's another thing to consider if you're using a laptop. All right, there's 26 of you. Good. All right, so that's everybody. So the correct answer there is A, protein synthesis. Ribosomes are responsible for protein synthesis. Another name for protein synthesis is translation. So we'll talk about transcription and translation later, but those two words can be used interchangeably. We call it translation because we're turning the RNA language into the protein language at the ribosome. So it's at the ribosome that takes place. B, manufacturing lipids, that would be the smooth endoplasmic reticulum. Cell movement and cell shape, that's the cytoskeleton. Packaging cell products for export, that's really the Golgi apparatus. And then energy production, that's the mitochondrion, but we haven't gotten there yet. All right, here, I'm going to skip that one. Okay, so mitochondria. Mitochondria are, their claim to fame is they're the energy producer inside the cell. You know, they're the power plant, and they're responsible for our need of oxygen, and they're responsible for our creation of CO2. So we all know that we breathe in oxygen and we breathe out CO2. Well, the reason for that is because of what the mitochondria do, and that is they take carbon compounds and using oxygen, they take the energy out of those carbon compounds, but they leave behind carbon dioxide, which we then have to get rid of. So mitochondria, you know, we could spend a whole day talking about these little guys, but they have a very complex double membrane structure. So they have, you know, an outer membrane, and then they have an inner membrane, and that inner membrane is kind of twisty and turny, a little bit like an endoplasmic reticulum is. The inner membrane surrounds what's called the matrix. So the inside of that inner membrane is the matrix. And it's where the matrix, or it's in the matrix that some of the fancier stuff that the mitochondria does actually happen. Mitochondria are also sort of unique. They have their own DNA and they replicate themselves. Now, one very common theory is that the mitochondria actually started as bacteria and then, you know, eons ago moved into our cells, and it's a synergy, you know, or a symbiosis, you know, because the cell provides the mitochondria with the food it likes to eat, and in exchange, the mitochondria produces ATP, which the cell then uses for all of its various cellular processes. So mitochondria are sort of unique like that, they have their own DNA, and in some ways, they behave almost like an independent organism. Now, they are under the control of the nucleus of the cell, though. So it's not like mitochondria can just divide whenever they want to. The nucleus controls that, but the mitochondria divide themselves with their own DNA that they have inside. So the short version of what takes place in the mitochondria is, well, we'll start out in the cytoplasm. So a nutrient like glucose is first broken down into two smaller pieces. So remember, glucose is a ring sugar, right, like this. Well, in the cytosol, we break two of those bonds. So we break the top one and bottom one, and now we're left with 2 3-carbon chains. And mitochondria love 3-carbon chains. That is their preferred, fuel or food source. So we call these pyruvate, each one of those. So the pyruvate gets fed into the mitochondria. There's a process called the citric acid cycle that takes place. And then at the end of that, CO2 is released and ATP is released. But we need to have oxygen in order to have this part of the system work. So just like I said, it's the mitochondria. This is cellular respiration, using oxygen to burn up carbon compounds to make CO2, ATP, and water. So that's primarily what the mitochondria does. And you all will spend much more time on the mitochondria in the next semester when we talk about energetics. But for now, just know that carbon compounds get burned with oxygen to make CO2, water, and ATP. All right. So in most cells, the most readily identifiable organelle is the nucleus. And we all learned in cell biology that the nucleus is sort of the brain or the control center of the cell. So how does that really work, though? Well, it's the nucleus that determines the long-term status of a cell. In other words, what kind of cell is it going to be? What is that cell going to do? What is its primary function going to be? Because the moment-to-moment and to some extent the day-to-day changes in the cell are not actually directed by the nucleus. They're directed out here in the cytoplasm by enzymes turning on and off, by receptor effects and membrane effects and all that. So what the nucleus ultimately does is it controls what happens in the cell by controlling what proteins get made. It's proteins that make everything that goes on in the cell happen. And the nucleus has all the recipes for all the proteins. and that's because that's DNA. DNA is recipes for proteins. So by controlling which proteins get made, turned on and turned off, the nucleus can control what happens out here in the cytosol. Because if it changes the proteins that are present out here, it's going to change what the cell can do. So therefore, it's going to change what the cell does. So that is how the nucleus controls things, is by controlling which proteins get made and then which proteins determine what that cell is going to do. All right. So a little anatomy of the nucleus. It's pretty simple. It has a double membrane here. There are holes in it called nuclear pores. It's through these holes that the RNA that comes out of the DNA inside actually goes out into the cytosol to get made into proteins. Many cells have what's called a nucleolus, a darker spot, either in the middle or off to the side. And the nucleolus is the area of DNA that's being actively used. We have, in every cell, we have a complete set of DNA. But every cell doesn't need all that. It only is going to use a tiny little fraction of all the DNA information that's present. So the nucleolus is sort of the working part of the library. The rest of the nucleus is the part of the library that's not in use. So if you use a real library as an example, the nucleolus is like the study room, and the rest of the nucleus are the racks where there's just book after book, floor after floor. So the nucleolus is where things are actually happening. So not all cells have a nucleus. Red blood cells, for example, when they're mature, have lost their nuclei. And some have as many as 5 or even more than 5 in large cells like muscle cells. All right. So because there's so much DNA in the nucleus, it has to be organized or it would just be a mess for the cell to work with. So most of the DNA, particularly the DNA that's not being used, is kept sort of tightly wound around itself and around some proteins called histones, and they're formed into chromatin. So chromatin is basically compressed DNA that is not currently being used. And then when the cell goes to divide, all that DNA has to be organized so that the cell can be sure that the daughter cell gets a whole set. You don't want to miss anything. You don't want to leave something out and then have that cell die because it doesn't have the protein recipe that it needs. So in the process of cell division, DNA is formed into what we call chromosomes, which are even tighter packed chunks of DNA than we see in the nucleus, usually in the histones. So each chromosome has a centromere, which is the center part of it, and this is the spot that the cell kind of grabs hold of in that process of cell division. So every time the cell divides, the chromosomes line up in the same way. So the DNA on one chromosome is consistent across all cells and across even most human beings anyway. So chromosomes are sort of an organized version of the DNA. So why is DNA so important? It's because of the information it contains. DNA is not really very chemically active. It doesn't really do anything interesting. Its primary role is to store information. And it does so using 4 letters, essentially, or 4 nucleotides, which we talked about last time. So A, T, G, and C. And each one of these has a match. So like G's always go with C's, T's always go with A's. So in the double strand of DNA, what makes that double strand happen is that complementarity, that T always matches with A and C always matches with G. Now, why did it evolve like this? Probably for stability and for error correction. Because each one of these has a complement, let's say this one right here got screwed up. Well, the DNA can fix itself because it knows that if there's a C on the one side, then there should be a G on the other. So it can repair itself because there's a duplication or a copy on the other side too. The process of turning this code into protein is called translation. And the way that happens, I know this is a review for at least most of you, is for every 3 letters, that means a particular amino acid. So like, and I don't have the code memorized, sorry, but let's say CTC meant glycine. So every time that there's this CTC, then the ribosome would go and pick a glycine to match that. So it's called the triplet code. So for every 3 letters, that's a signal to the ribosome of which amino acid should come next in that protein chain. All right. So, we've got sort of a logistics problem here because the recipe for the protein is in the nucleus, but the protein machine and manufacturing system is out there in the cytosol. So we need a way to get that information from the nucleus out into the cytosol. So that is where RNA comes in. RNA, messenger RNA specifically, it forms a copy of a chunk of DNA, and then it takes that information and the RNA goes out of the nucleus into the cytoplasm, finds a ribosome, and then creates that protein using that model. So DNA has the recipe, RNA takes that recipe out, and then the ribosome turns that recipe into a protein. So the process of making an RNA molecule from a DNA template, that's called transcription. So we've got 2 fancy words here, transcription and translation. Transcription is making an RNA from a DNA template. Translation is making a protein from an RNA template. So we have 2 different words here. And I have a movie to show you. Okay. I am going to turn the lights off for this part. Please don't go to sleep. All right. I answered the volume up a little. Okay. So this is transcription and translation.
With computer animation, we can enter the cell to view this remarkable system at work.
Because it's really cool.
After entering the heart of the cell, we see the tightly wound strands of DNA, storehouses for the instructions necessary to build every protein in an organism. In a process known as transcription, a molecular machine first unwinds a section of the DNA helix to expose the genetic instructions needed to assemble a specific protein molecule. Another machine then copies these instructions to form a molecule known as messenger RNA.
Isn't that cool? Like a zipper.
When transcription is complete, the slender RNA strand carries the genetic information through the nuclear pore complex, the gatekeeper for traffic in and out of the cell nucleus. The messenger RNA strand is directed to a 2-part molecular factory called a ribosome. After attaching itself securely, the process of translation begins. Inside the ribosome, a molecular assembly line builds a specifically sequenced chain of amino acids. These amino acids are transported from...
So we've got the messenger RNA up here, and this is called tRNA, or transfer RNA, and it has the triplet code that I was talking about that goes with a particular amino acid. And then what you're seeing down here is this is the protein that's being made, the primary structure, the order of amino acids. So we're going from DNA code, or that went to RNA code, and now we're translating it into an actual protein that'll do something.
Other parts of the cell, and then linked into chains often hundreds of units long. Their sequential arrangement determines the type of protein manufactured. When the chain is finished, it is moved from the ribosome to a barrel-shaped machine that helps fold it into the precise shape critical to its function. After the chain is folded into a protein, it is then released and shepherded by another molecular machine to the exact location where it is needed.
So it's all about shape. So that's transcription and translation. All right. And done very well, I think. All right, we'll end with a question. All right, the nucleus of a cell functions to do which of those things? All right, jump in there, last two people. All right, I'm going to call it early because almost all the answers are in. Okay, so the answer is D, control and coordinate cellular activities, particularly in the long-term aspect, not necessarily the short-term, because the cytosol can do a lot of fancy turning things on and off, but it's the nucleus that controls what that cell's going to do over its life. All right, thank you, everybody. Have a good weekend, and we'll jump back